Hello everybody, and welcome to today's webinar, entitled Fluorescence and Compensation in Flow Cytometry. Your host will be Dr. Mike Blundell, one of our product managers here at Bio-Rad. This is the third in our series of flow cytometry webinars, with more to follow. Mike graduated from the University of Edinburgh with a BSc in Immunology. He then moved to University College London, where he joined the group of Professor Adrian Thrasher and obtained his PhD. His thesis research was focused on the primary immunodeficiency Wiskott-Aldrich syndrome. He investigated novel activating mutations and developed lentiviral vectors for use in gene therapy treatments, some of which have now been used in clinical trials. Mike contributed to 50 peer-reviewed papers and has now left academia to become a product manager for flow cytometry. As we're trying to make this webinar as interactive as possible, please submit questions or comments via the Q&A box located at the bottom of the screen. We will try to answer as many questions as possible in the session following the main presentation. Should any questions remain unanswered, we will reply by email. The webinar will be recorded and an on-demand version will be available shortly. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide, which describes solutions for the most common technical problems. In addition to webinars, we regularly post blog articles, not just on flow cytometry, but other applications and research areas. We also have blog posts on current scientific discussions and the latest biomedical discoveries. Please feel free to check out the blog and comment on one of the many articles. In addition, you can follow us on social media to stay up to date on antibody resources, news and products. Our social media channels can be accessed via the icons at the bottom of the screen. During the webinar, you can interact with us on Twitter by submitting questions or webinar feedback using the hashtag BioRadWebinars. I hope you will enjoy the webinar, and now over to you, Mike.
Hello everyone. Welcome to this flow cytometry webinar on the subject of fluorescence and its use in flow cytometry. As I'm sure you're aware, light is a form of electromagnetic energy that travels in waves. The human eye can detect wavelengths between 390 nanometers and 700 and are observed as varying colours. Below this is ultraviolet and above is infrared light. Shorter wavelengths have more energy than longer wavelengths of light. Flow cytometry relies on fluorophores attached to antibodies to detect markers of interest both from the cell surface and intracellular. Fluorophores are molecules that accept light energy at a given wavelength and rapidly emit this light at a longer wavelength. In flow cytometry, the light source is most often a laser, or now more commonly multiple lasers. The filters and detectors, called PMTs, in a flow cytometer will allow light from around 380 to over 800 nanometers to be detected. When a fluorophore, such as fluorescine, which was the first fluorophore to be commonly used in immunofluorescence, absorbs light, its electrons become excited and move from a resting state, shown as S0 in this simple Jablonski diagram, to a maximal energy level called the electronic singlet state, shown as S2 in this diagram. The amount of energy required for this transition and the length of time spent in this state will differ for each fluorophore. The fluorophore undergoes a conformational change. The electron falls to a lower, more stable energy level called the singlet state, shown as S1, with some of the energy released as heat. The electron subsequently falls back to its resting state, releasing the remaining energy as fluorescence. This cycle lasts nanoseconds and can be repeated many thousand times, thus allowing recycling and amplification of signal. The emitted light typically contains less energy than what was put in to excite it. Therefore, the emission wavelength of any fluorophore is generally longer than its excitation wavelength. Although fluorophores will absorb and emit a range of light wavelengths, which we will discuss in more detail later, they have an excitation maximum and emission maximum. The excitation max A and emission max C can be seen here for fluorescine. The difference between these two values, shown here as B, is called Stokes shift, and it is this property that gives fluorophores their use in flow cytometry. I have shown here a table of common fluorophores with their excitation and emission maxima and their relative brightness on a scale of 1 to 5, with 5 being the brightest. As can be seen, fluorophores are not all the same, even if they have similar excitation and emission spectra. Furthermore, some fluorophores are specialized for use in flow cytometry, such as PE, as it is very bright, but can be easily photo-bleached. Fluorophores such as APC and Alexa 647 have similar excitation and emission spectra, but APC is not suitable for use in microscopy, as it too photo-bleaches easily, whereas Alexa 647 is more photostable. Finally, fluorophores such as FITC or Alexa 488 have similar excitation and emission profiles and can be both used in many fluorescence-based antibody applications. For more information on fluorophores, go to www.bioradantibodies.com/fluorophores. A quick example of the differences between fluorophores is shown in this slide. CD3 was conjugated to 2 different fluorophores using ReadyLink kits from Bio-Rad, and peripheral blood was stained. The positive and negative populations can be clearly identified for both stains, but the distance between the positive and negative, marked on the plots as R1, is clearly different, with the brighter dye being the red laser excitable 700-713 dye. The brightness of a fluorophore is important if the antigen or cells being detected are not abundant, and is important for signal resolution when assembling larger multicolor panels. In addition to single molecule fluorophores, tandem fluorophores are a clever way of increasing the Stokes shift to allow excitation and detection of more fluorophores excited from a single laser. They work by covalently linking a donor fluorophore, typically PE or APC, to an acceptor fluorophore that has a longer maximal fluorescence emission, such as Alexa 647. When the donor dye is excited, it transfers its energy to the acceptor dye in a process called fluorescence resonance energy transfer. So, if we use PE and 8PE Alexa 647, as shown on this graph, although both are excited by a 561 nanometer laser, instead of emitting at 578 nanometers, as can be seen for PE on the graph, PE Alexa 647 emits at 667 nanometers. Tandem fluorophores, however, may need special handling, as they can be sensitive to light and some fixation protocols. For more information, go to www.bioradantibodies.com/fluorophores. So as I mentioned, although fluorophores have a maximal emission wavelength, they will actually emit over a wider wavelength. And this can be seen here for FITC in green and PE in yellow. The best signal for FITC will be obtained by having a detector after a filter around about 520 nanometers. However, you will still get a signal, albeit weaker, in the PE detector, which is located after a filter around about 580 nanometers. The proportion of fluorescence from FITC that can be detected in the PE channel is marked in red. Conversely, the proportion of PE fluorescence that can be detected in the FITC channel is marked in blue. And this is called spillover, and has to be removed by a process called compensation. Here, using a spectroviewer, which is part of the Everest software, free with the ZE5, the new cell analyzer from Bio-Rad, you can see where spillover of a fluorophore from a laser may occur, highlighted here by the yellow and orange circles. So how does this actually look on a flow cytometer? Well, the dot plots here show the fluorescence detected in the PE channel as predicted in the previous slide, and the fluorescence in the PE Cy5 channel, even though the sample has only been stained with FITC. As I mentioned, the method to remove this unwanted fluorescence spillover is to apply compensation. Compensation is simply a mathematical algorithm that allows you to separate and remove this unwanted fluorescence. Here we can see FITC and PE single staining with no compensation. When compensated correctly in the direction of the arrows, you can remove the unwanted fluorescence, and the true level of staining is revealed. Without compensation, in this case, cells from human peripheral blood would appear double positive for CD4 and CD19, which are found on T and B cells respectively, when in fact they are actually singly positive for each marker. Compensation can be applied automatically using software at acquisition by acquiring singly stained samples, or it can be applied after acquisition in many analysis software packages, either automatically or manually, although many experts recommend using the computer software to avoid bias. This slide, taken from my recent flow cytometry controls webinar, shows what controls you need to accurately apply compensation. Compensation controls are samples singly stained for a fluorophore, so you can detect the spillover into other channels. Quickly to recap though, there are some important things to consider when using single stain samples. Firstly, the fluorophore needs to be the same and its brightness similar to the one used in your experiment. You also need to collect enough events to accurately apply the compensation and be statistically significant. Finally, each fluorophore should be compensated against every other fluorophore used. Therefore, single stains will be required for every fluorophore used. For more information on all flow cytometry controls, go to www.bioradantibodies.com/FC controls. The reason for checking all the other channels for fluorescence, including those from other lasers, and applying compensation with single stain controls is shown in this rather busy slide. Human peripheral blood was stained with CD4PE ElectroFluor 647, and using the Z5 SpectraViewer, we can predict the potential fluorescence from spillover from the 561 nanometer laser and from other lasers due to cross-laser excitation. As can be seen here, although PE ElectroFluor 647 is optimally excited by the 561 nanometer laser, there is also some excitation of either PE or ElectroFluor 647, the two fluorophores found in the Tandem die, by all the other lasers, and can be detected at both 578 nanometers, the emission of PE, and at 665 nanometers, the emission of ElectroFluor 647, depending on which laser is exciting the fluorophore. So what does this spillover actually look like? Well, the dot plot shown here of PE ElectroFluor 647 on the x-axis, plotted against all the other channels on the y-axis, highlighted by the circles, show the actual amount of cross-laser excitation and the resulting unwanted fluorescence visible in the other channels before compensation has been applied. Fluorescence spillover and cross-laser excitation will be a problem if you intend to use these filter sets to collect fluorescence from other fluorophores, and compensation will need to be applied. There are ways to avoid fluorescence spillover and therefore compensation, especially when there are a few markers to be investigated. The easiest way is to separate the fluorophores across the different lasers and filters on your instrument. The more lasers and filters available, for example the Z5 from Bio-Rad has 5 lasers and 30 parameters, the easier this is to do. An example of this is shown here, where a simple 4-color panel can be put together using Pacific Blue, FITSI, PE ElectroFluor 750 and Alexa 647 on a 3-laser instrument. As can be seen, although there is some spillover and cross-laser excitation, the filters used to collect fluorescence are not located where this spillover and cross-laser excitation occurs. Therefore, there is no need for compensation. So to help you with your flow cytometry, we have actually put together 13 4-color immunophenotyping panels for human peripheral blood that require no compensation. We have included images to help show staining patterns for the populations for easy identification. Panels include identification of T-cell populations, such as memory and naive, helper, cytotoxic, and regulatory cells. It also includes B-cell populations, such as class-switched, memory, and naive, and finally, NK cell and myeloid cell populations. These panels are useful both in identification of common blood cells or as a starting point for building your own panels, where markers can be switched or extra markers added to suit your experiments. To find out more, go to www.bioradantibodies.com/no-comp-panels. If you want to increase the size of the panels and still avoid compensation, it's often easiest to look at where there are gaps in the instrument detectors and pick fluorophores that are compatible. If we start with the same 4-color panel I showed earlier, but have split the fluorophores across 4 lasers, so a PE Alexa 750 is excited using the 561 nanometer laser, we can look for the gaps. One example could be Percy-P, which is excited by the 488 nanometer laser, and though it has some cross-laser excitation, they are in areas that do not interfere with the current 4-color panel. Additional gaps can be found on the violet laser at around 615 nanometers, and if you have an instrument with an ultraviolet laser, there will be gaps there too. This means, by careful choice of fluorophores, with a 5-laser instrument, 7 and possibly 8 color panels can be built that will require little or no compensation. A lot of the time, due to limitations in cytometer configuration or availability of antibodies with fluorophores to fit these gaps, compensation is unavoidable. This should not be seen as a problem, however. I have built this 8-color panel of CD3, CD4, CD8, CD19, CD20, CD14 and CD66B, including a live dead stain, to look at human blood leukocytes. Fluorophores have been chosen across lasers and filters, as described earlier with the smaller panel, but they're not completely separated. There is some spillover from single laser excitation of fluorophores, as can be seen for Alexa 647 into Alexa 700, and there's some cross-laser excitation, as can be seen from PE Sci5 into the PE and Alexa 647 channel. However, by following the rules for compensation and including the right controls described earlier, having a single color stain sample for each that has 2 clear populations and compensating each channel you are using, you can still get high quality staining. And that is shown here on this slide. We are showing the staining obtained from the 8-color panel described on the previous slide with the data obtained on the Z5 cell analyzer. After dead cell exclusion, CD14 positive monocyte and CD66B positive granulocyte populations can be identified in the myeloid gate. And in the lymphoid gate, CD3 negative, CD19, CD20 double positive B cells can be identified. Also in the lymphoid gate, CD3 positive T cells can be identified with helper and cytotoxic T cell populations also revealed by the CD4 and CD8 staining. There are several additional rules to building larger multicolor flow cytometry panels, and I will briefly mention them today, but we'll have a webinar later in the year to discuss building multicolor panels in more detail. In addition to the fluorophore properties which we have discussed today, one of the first things to think about is the density of your antigen. Low density antigens and rare cells require bright fluorophores, and higher density antigens and more abundant cells can use dimmer fluorophores. The marker expression patterns on your cells can also help you choose your fluorophores. Mutually exclusive markers can be used for closely overlapping fluorophores, as you know they should not co-express, making compensation straightforward. Similarly, the parent-descendant rule, for example as seen for CD3 and CD4, can also be used for closely overlapping fluorophores, as expression on both is expected and therefore not a problem. For unknown markers, markers with continuous expression, and activation markers, it is best to avoid closely overlapping fluorophores. A dump channel, where several populations are stained with antibodies, just with one fluorophore, can also remove unwanted background staining, helping clean up the analysis. Finally, titration of antibodies will optimize the positive signal over the background and reduce the background staining, whilst potentially reducing the amount of antibody required, saving you money. For more information on flow cytometry, including building multicolor panels, controls, protocols, fluorophore charts, gating strategies, and an updated flow cytometry guide covering the basics of flow cytometry, go to www.bioradantibodies.com/resources. There you will also find resources for other antibody-based applications, such as Western blot, IHC, ELISA, and immunofluorescence. In addition to this flow cytometry webinar, in our resources section we have 2 other flow cytometry webinars, including one on controls, essential for all flow experiments. We also have application-based webinars on topics such as mastering IHC and immunoprecipitation. And we have research area webinars covering areas such as apoptosis, all available to view on demand. The last slide today is to inform you of all the antibodies, reagents, controls and instruments we have here at Bio-Rad to help you with your flow cytometry experiments. We have antibodies, both single unit and cocktails, with a range of fluorophores. We have viability dyes, proliferation dyes, and easy-to-use apoptosis kits. Go to bioradantibodies.com/flow for more details. We also have an automated cell sorter, the S3E, and a new cell analyzer, the ZE5, which I'm going to spend a minute telling you about. This new innovative cytometer has the flexibility to meet a broad range of needs, whether it is 2 parameters or 28, from single tubes to 384 well plates. The ZE5 is accessible for novice flow cytometry users with a fluorophore selector, a spectroviewer which shows fluorophore spillover, which I've showed you today, and quick action buttons to allow selection of the next setup step with ease. And yet it's flexible enough for the more experienced users, with extra forward scatter detector for small particles, absolute counting without beads, and the ability to add reagents to your samples in real time for kinetic experiments. And that brings me to the end of today's webinar. I'd like to thank you for listening.
Thank you, Mike, for that interesting and informative presentation. And now I would like to open the Q&A session. I'm aware that some of you might have to leave us early. If you're unable to stay with us for the duration of the Q&A session or missed your chance to submit a question, you can contact us or our technical support team by email. We offer technical support in English, French and German, and you can simply call us or send us an email. For more information about how to get in touch with our technical support team, please visit our website, bioradantibodies.com/technical. For the Q&A session, we are joined by another member of our team, Dr. Steve Clasper. Steve holds a PhD in biochemistry from the University of Liverpool and has wide-ranging experience in antibody applications that he has gained in both academia and industry. And now, over to you, Steve.
Thanks, Katie. Thanks, Mike. I'm Steve. I've been looking over the questions that have been coming in during Mike's webinar, and we've got some that are quite general and a few that are more specific, so we'll do a bit of a mixture. The first question is, once I've set up compensation for a panel, can I use these values for future experiments? Well, strictly speaking, you should always perform compensation when a panel is run. Quite often it's tempting just to not do this step and use old values. And if your cytometer can self-calibrate at the start of every session to automatically adjust the internal voltages, this takes into account any change in laser performance over time, so you may actually be able to reuse a compensation matrix. We don't really recommend this for important data though, and there are a few caveats if you do this. Firstly, the PMT voltages have to remain at the same values used in the original experiment. Also, you need to use identical fluorophores, and the brightness levels mustn't exceed those that were originally used to set the compensation. If you're using tandem dyes in your panel, then these can actually optically uncouple over time, and this will increase spillover. I would recommend you always perform a compensation step with panels that include these fluorophores. The next question is, what is the difference between using cells or beads for compensation? Is one better than the other? And can you use a mix of beads and cells to generate a compensation matrix for a staining panel? Well, there's no difference between using cells or beads with regards to accuracy. Both have different benefits. Beads are ideal if you have a limited sample, so you can use all the cells in the experiment. If you have a low abundant antigen, then using beads would be advantageous to get a larger positive population. The problem with beads, however, is you can't use these for dyes, so in this case, you'll have to use cells for things like live dead staining. But as long as the positive and negative populations are the same for each control, you can actually use a mixture of both beads and cells for your compensation panel. For compensation samples, can I use a different antibody conjugated to the same fluorophore if my antigen of interest is of low expression? Well, yes, you can do this, with the exception of tandem dyes. The problem here is the degree of initial conjugation and the breakdown over time will vary between antibodies. So the option here is to use beads. The next question is quite a specific one about a problem someone's having. When I use propidium iodide as a live dead stain in the PE-Cy5 channel, I seem to lose a lot of PE-positive staining compared to cells that are not stained for viability. Is the compensation to blame? Well, this probably is a compensation problem. If the compensation between the PE and the PI is not adequate, your PE-positive cells will appear to be dead in your viability gate, and so you'll end up excluding them from your analysis. So always ensure that you use true single-stain tubes for setting up compensation. If you're using cells rather than beads for the single colours, then this means you must exclude the viability stain from these tubes. The next question is it possible to compensate manually with double staining, or can you only do this for single staining? Well, you can only compensate with single staining because when you need to use a single fluorophore to measure the spillover into each empty channel. If you use double stain cells, it's going to skew the results and produce the wrong values. The next question is, can compensation controls be too bright? In fact, you'll often find you get the opposite problem, is that controls are too dim. They need to be as bright or as brighter than the brightest signal in the sample. The fluorophore may not display measurable spillover into any of the correct channels if it's dim, so if your compensation controls aren't bright enough, then you may get undercompensation. Someone sent in a question asking that, about the built-in compensation function in their analysis software. Should they use it? And we would say yes, definitely, that the software is much better at applying the correct values for compensation than if you try to set these by eye. This is assuming you follow the correct rules for single staining. When you set the values by eye using log plots, it's quite often easy to end up undercompensating. I think the last question we have time for now is, can I use the same compensation settings for fluorophores that use the same filter set? And the example we've been sent is APC and Fluor 647. When we first look at this, it seems logical that two fluorophores that are excited by the same laser and emitting the same filter should share the same compensation values. However, APC and Fluor 647 are quite different molecules. APC is a protein, whilst Fluor 647 is a synthetic dye. And if you look at the excitation and emission spectra for these, you see that they don't overlap entirely. Both are excited by the 640 nanometer laser. If you look at APC, it shows more excitement by, for example, the 561 nanometer laser than Fluor 647. And it's therefore going to need more compensation against some PE tandems, for example. So it's very important to set the compensation for each fluorophore independently to prevent inaccuracies, even if they appear to be the same. Okay, well, thanks for listening to this webinar. If you want any more ideas about compensation, please look at the flow guide from the website. I hope you find that compensation isn't as daunting as it first appears. Just have a go with your single staining and have a look at some of the no compensation panels on the website as well. Thanks for listening.